My name is Yanga Zembe. I'm an associate professor of community development in the School of Built Environment and Development Studies. I'm also the school's academic leader for community engagement. And it has been inspiring and amazing and wonderful to be here. I'm here to chair the session that we're going to be having now. And I'm going to introduce to us the academic and speaker who's going to be presenting to us what I am sure is going to be an inspiring talk. So the person that I'm introducing is Professor Nceba Gqaleni, who is my umkhaya. That means we come from the same, originally, from the same province, the Eastern Cape. He is a professor in traditional medicine in the faculty Africa Health Research Institute. I had time that I spent with him last week, and he just blew me away. He is a pioneer. He is amongst the first in this country to truly pioneer the decolonization of not just education, but of an entire discipline, in this case, the discipline of medicine. He sits on various august bodies, nationally and internationally. He's been on various presidential task teams in this country advising on traditional medicine. He also is a member of the WHO expert regional panel on COVID-19 and traditional medicine. Professor Gqaleni, as I've said, has a long history of doing incredible work to Africanize the discipline of medicine, but also to Africanize the way we do research, the way we think about research. He's really a textbook example of how you take the world to Africa and bring Africa to the world. He has done a lot of training on how to integrate traditional medicine within the public health curriculum. At a time in this country, I think in the early 2000s, where we were grappling with the AIDS pandemic, I'm now in community development, but I'm actually a public health scientist. I still remember that time where we were grappling with AIDS. We had an unbelievably high mortality rate. Our behavior change interventions weren't working. And we were focusing entirely on this Western biomedical model to help us. And Prof. Gqaleni was going where angels fear to tread, because he was actually at that time, in that time of crisis, saying, there is an African solution, there could be an African solution, we should be considering an African solution to the problem of AIDS. And we talked last week about how tough that was, because at the time, whenever we heard that people who had developed full-blown AIDS were consulting traditional doctors, were going to Izangoma and such, those of us in public health would say, oh no, you are going to die. There was no room, there was no place in the room for an African traditional medical intervention. And so he faced a lot of backlash. Many, many doors were closed, couldn't do research, particularly clinical trials, struggled to have laboratories opened so he could test African traditional medicines and African traditional herbs that could possibly treat AIDS. And he pushed, and in fact then his incredible work of just staying focused and being driven by this vision to bring an Afrocentric solution to our public health problems, pushed him such that he was able to establish a center whose work entirely focused on the use of traditional medicines to treat those diseases that are most prominent in our burden of disease in South Africa. So he built a huge body of work, cementing traditional healing and traditional medicine as an academic discipline. As I've said before, to me, he really is a textbook example of how, not just how to do internationalization, but why we need it, why it works, why we should take the opportunities that are available to us by globalization. He is going to talk to us now about how the COVID pandemic has opened up spaces for traditional medicine to take center stage, to become one of the leading interventions that we tend to in the treatment of COVID-19. And he's going to talk about how partnerships with international collaborators at a time where in South Africa, scientists, basic research scientists, virologists would not open doors to a scientist wanting to explore the efficacy of traditional medicine in the treatment of diseases. How, at that time when those doors closed locally, how it is international partners, universities and institutions across our seas that opened those doors for him and made it possible for him to build the kind of infrastructure that he needed to be able to advance the science of traditional medicine. So we are going to hear, as I said, what I believe is going to be truly inspirational, and we're going to hear about how we can use each other, how we can use connections that we build beyond our borders, even when our own doors at home are closed, to advance science. And we're going to hear about how to enter collaborations with not just the purpose of meeting our research objectives, but the purpose also of ensuring that a distinct African identity is infused in the way we work. I think Lawrence talked about this, how a lot of times when we speak international, even in your head, you imagine, you may immediately think overseas, USA. You know, you don't think that international incorporates and includes very much the countries around us within the continent of Africa. And often we enter those partnerships and we entering to assimilate, to absorb, rather than to contribute and spearhead. And Professor Gqaleni has shown us and is going to tell us how he goes there and he brings African solutions to the world. And he says, Africa is a solution, we're coming with an African curriculum on how to treat diseases such as COVID-19. Over to you, Prof. Gqaleni. Prof. Gqaleni, you'll have about 20 minutes, and then I will stop and take questions. Thank you.
Thank you, Program Director, for those kind words of introduction. You are equally an inspiration because it was great to interact with you before this plenary. I think it's still good morning, good day, ladies and gentlemen, and I'm grateful really to the organizers. I normally call her the Zondo Commission. When they call you, you can't refuse. When she calls you, you can't say no. So I'll suffer them sholozi. Otherwise. But thank you. I coined the title of my talk. I'm hopefully, I don't know how to tell stories like social scientists do. I'm a natural scientist, but I will try and just carve the path. I started as a biochemist, teaching medical students the subjects they didn't like in second year because they were still keen to get onto patients, and I'm busy telling them about the pathways, the biochemical pathways, boring. So I never started to be a researcher on traditional medicine. Traditional medicine found me, and it never left me, and if I use the word. In year 2000, we had a new dean at the Nelson Mandela School of Medicine with a strategy plan that followed. We realized that for the medical school to grow, which was at the time established 50 years ago to train about 20 black doctors. In 1950, when it was opened, the med school had grown to take in 2000 about 200 undergraduates a year after a 6-year program. And of course, they had a similar number of registrars per year in the 4-year training program. So we did not have spaces to grow and things like that. But one of the things we discussed was that we need 3 areas that we need to do to decant these buildings, to create new labs, and that's how molecular medicine, and now you see COVID, we are excelling in that. In the year 2000, we decided on a strategy that molecular medicine should be the future for us. Secondly, telemedicine. You've heard from Mr. Appleton that even Durban itself is 68% rural. The city is a deceiver. So telemedicine became our next strategy. The third was traditional medicine. The unfairness of the patients who choose to consult traditional healers and come to biomedicine and get ridiculous treatment just because they chosen to go to a healer in our health centers. So we needed to reorient our students to that, let alone the teachers. That became a motherless program. And the telemedicine moved, modern molecular medicine moved, until I was roped in 2003 to spearhead this. And I negotiated an agreement with the leadership of traditional healers in this province with the medical school. When I took it to my dean, we had already kind of mapped an MOU, translated into Zulu, and I gave it to him. And he said, hey, no medical school has ever done this. This is big. So we must invite His Majesty, the Minister of Health, the consulates in around Durban came. It was a massive event in the Steve Biko lecture theater of the medical school. You know, it was a game changer in a way, because for that, the medical school to bring the king to them, it meant you are serious in what we want to do. You would not invoke the king. Maybe the health minister is a politician, but the king is not. So that's how it started. Of course, that work attracted, I didn't expect, to be honest. I was just doing a little thing, but it opened doors. Governments across were keen on this program. They funded us, and suddenly we had one of the, competing with the big wigs in the medical school in terms of HIV/AIDS funding, and I was nothing. I was like, who's this one? And here we are. And the traditional healers respected and trusted that relationship with me. And so our intention was training, teaching students. And now here is research. Healers come with their products. Hey, prof, I've got this thing. I was not even prof then. Hey, doc, I have this thing. And so I was not a virologist. It was not my competence. I was a biochemist. So those spaces that had infrastructure to do virology and stuff like that, they had their different thinking. They had their different objectives. So it became uneasy to then take this sacred medicine that people want to have evaluated for its possible contribution to HIV. And that's when it became a challenge, an opportunity, yet a challenge. And that's when it became obvious to me that we've got to look elsewhere, not in South Africa. And so the first project we had, it was on Kaposi sarcoma, which was an AIDS-defining cancer at the time. And this is a patient who had Kaposi sarcoma. He was not taking his doctor's prescription, but he was going to his doctor for follow-ups because he was on medical aid. But this doctor sees that, no, man, the response here is not what I'm expecting. What are you doing? The patient tells him and says, look, I'm not a researcher. Find people who can help you. And that's how we came across Ubabu Kumalo. And we don't have models to study this, and we had collaboration with the University of Florida. We sent a PhD student there, Untitled Chapter, who spent 3.5 years there doing work. And what this work for us, it was the first, a scientific breakthrough in a sense, that When the virus infects the cell, of course, it gets its DNA into the human genome. The cell is transformed to cancerous. So when you treat the cancer, you're not treating the causative agent. But what this medicine does can you go move to the next slide? Next. What it's doing, it's basically, you can see on the right, the cells are intact, and then on the right, it lyses the cell, the DNA is split up, but what's happened is that the human DNA, as part of apoptosis, collapses, but the viral genome stays. And what it's doing, the next slide, what it's doing, you can see that the viruses, the viral copies, actually it's a dose response. You don't need a huge dose of this medicine for that. So now you can treat the causative agent of the cancer and the cancer itself. There will be no relapse. What is $1 million question for postdocs who want to do this? What is this? How does this happen that this medicine makes a separation between a human genome and a viral genome? We don't know. It's a question we want to, maybe that will be for Nature publication in future. Next. So that was the first challenge for us. And the second then became, I see it's gone. We had approached by a professor from Wits, who was sent to us by the Department of Health. He had, not sure how he had got to know Mr. Ngabinde from Dundee, but he had been treating patients. Can go to the next slide. He had been, let's leave it there. He had been treating patients. He comes from a long lineage of herbalists, of families, and they had a medicine they were using for syphilis. So when AIDS struck, he realized that this is a sexually transmitted infection. Let me give the medicines of my grandfathers. And he began to see results. How he landed at Wits, I don't know. But this professor's wife, Professor Stewart, got cancer. And they needed to go back to UK to be cared under the NHLS. But he said, I want to leave this medicine in the hands of good men. I can't leave it with anyone. So then, that's when I got approached by the Department of Health to do this work. Now, next slide. What this medicine is doing, this is work of our PhD students. Go to the, yes. Yes. These are virus genomes these are viruses that are in South Africa, floating around, type C viruses. And we can see that we can achieve up to 94% clearance of the virus in vitro. Next slide. We can also compare it to, this is a pure compound in blue, equivalence, a standard antiretroviral. So it's pure, so it gives you a clear sigmoid curve, and you can see here on the other side. But then we got stuck. Go to the next slide. What we were seeing is that, of course, there is the killing of the virus in blue. But in this blue, there's a stimulation, which was a kind of puzzling our heads. And we knew that antiretrovirals don't cure the virus. They clear it in the blood, but the virus hides in tissue and relays there latently so. And so we could not find a lab that could do this latency testing for us. Until I was teaching a class at a program of SANTE with the Africa Health Research Institute, where they had a program of students from Canada who spent a few months doing HIV forecast. So I gave a lecture to them, and I showed one of these slides, and they said, I have a professor in my university, Simon Fraser University in Canada, in Vancouver. Can I talk to him about this work? He's got a model for latency. This was a PhD student working in his lab. Said yes. That night, because of the time zones, at night she sent a message to her professor and introduced us, and we connected. We met in Kenya in a conference, sent him samples, and this is what he showed. Next slide. What he showed, the first is the fact that we had different, I work with a chemist at the University of Pretoria who likes to put solvents and things. But what we found was that with increased differences in solvents, you can increase the potency of this medicine. But there was something when he used dichloromethane that he found is a trend, it goes against the trend. And we were asking, could that be telling us about potent latency reversal of latent HIV? Next slide. And that's what he did from his test. So now we know we have a product that is possibly more potent than efavirenz, because the efavirenz is a pure compound. This is a crude extract. If we were to further purify these compounds, which we know, so we know we have a blockbuster. And now it reverses latent HIV. So now we know we have a package for cure. We're getting ready now to do a phase 1 clinical trial, and we're going to go and do a phase 2 clinical trial. We don't know how the pharmaceutical industry is going to respond to us, because this is a disruptor, so to say. And we're quite happy. We'll stand up to that challenge. Now, next. One of the things that we funded by the Department of Science and Innovation, I chair the consortium, is to also develop what we call nutraceuticals, food that are medicine. And one product is moringa that people throw around, but not only solid evidence that goes with it. And so we had a student from Nigeria, a PhD student, Aisha Gambo, and so she took this, did a PhD in Nigeria, in a setting like ours, a rural setting where people live on pap and fleece in KZN. I don't know what they live it in Nigeria, is a swallow, whatever they swallow, they call it. So we then supplemented the food 3 times a day, moringa. And we had a control, which was starch dyed to look like. So we had 2 control arms on this. Next. Why did we do this in Nigeria, not in South Africa? Maybe I should have said that. There are also politics here of regulation. Our SAPRA, or the Medicines Control Council, the first clinical trial we did in South Africa, took us 3.5 years to approve. We had a 5-year grant from the NIH. 3.5 years to approve. And when they approved, they changed our entry points, recruitment criteria. In the 3 years, South Africa, for access to antiretrovirals, you needed to have a CD4 of below 200. It then changed in that period of three years to less than 350. For us, they said we must recruit patients with CD4 above 550. What that means, we had already designed a study, we had budgeted for it. Now we were to recruit a sample of about 400, 200 each arm. We needed about 3,000 or 5,000 to see small changes. So we had a trial. We could only prove safety, but we could not prove efficacy because the boundaries were so wide. These are healthy subjects, so some will come with CD4 900, CD4 800. To show small differences was just impossible. So we tried this now in Nigeria. Nigeria they're probably an African country better than us. So we ran a clinical trial there. Next slide. And of course, there's a complex statistics was something I had to know about clinical trials. Next. But what we can show clearly is that the people who are on supplementing with moringa had a rise in CD4 counts much better than the people who are taking their normal diet. This was a 6-month study, closely followed up in terms of their daily food that they were eating. But the difference was moringa that this is moringa from South Africa. But it's a plant that you don't need to buy moringa, we can grow it and eat it in your home. So it's food as medicine, that's what you say. But at least we are showing evidence, which is important. And these are collaboration. So we had a university in Nigeria that now accepted to lead this clinical trial in working with the student. COVID comes, the president of Madagascar creates a storm in a teacup, but it was a big storm for South Africa because the Minister of Health of Madagascar, see, when a president makes an announcement, journalists are asking for scientific evidence. The president does not know all of those things. And other presidents bring aeroplane to take orders and take to their countries. Journalists are still asking scientists. I was actually interviewed myself. What do you say about this? I said, I don't know. He's a president. So they invite South Africa to help, but it became problematic. How will, if a president announces, is it going to now the president of South Africa writing the president of Madagascar? It became a diplomatic nightmare. So eventually our ministers asked us scientists in South Africa to say, let's look at our own. Now that Madagascar started it was good for us, in a sense, a blessing in disguise. But then we had lockdowns. I'm not a virologist. How am I going to do this? How are we going to do this in our consortium? Even CSR at the time, they had not yet developed models for COVID at that time. Thanks for collaborators. Again, we had a friend at the Worcester Institute in the States. Next slide. Who started? So the objective for us as a consortium I was chair of is to then look at all the indigenous plants that are known to be used for respiratory or viral diseases of this nature, produce quantities that we can do testing on, and then, of course, then do in vitro test on this stuff. And next slide, I'm not going to go to you are not an audience to deal with this, but one of the things we knew is that there's this ACE2 receptor that the virus enters through into the human cell. So we needed to see, can we disrupt that? And of course, to see what is the toxicity, what is the IC50 of this extract. And a colleague of mine, Ian at THN at the Worcester, fortunately for them, they had permits to go to the labs and start if it was COVID related. It came later for us. And this is a model that is used. Next slide. And these are the results. They're not presented in the most fancy of ways, but these are plant extracts. We know them, although we're not telling you what their names are, because what happened with Artemisia? People started going to harvest any plant in South Africa. Any Artemisia was in a chemist, they were gone. So we don't call names now. So, but what's the good thing here is that we're finding something that is promising. These extracts can disrupt this ACE2 complex, and you can see the selectivity is low for these extracts. We also looked for our own Artemisia, South African Artemisia afra. Next, we couldn't find activity, so to say. And we also found part of product Nkabinde that we're working with, and you can see that we could, from one of the plants of the mixture, they had about 116, close to 120% selectivity. So at least we know now in terms of how to fish for these things. And of course, next, of course, in this work you do, medicine is about balancing toxicity and efficacy. So we always have to check these two as we do this work. Next. So at least now we knew that we do have plants that we can work with. I've since hired a technician at ARI who's actually sitting at the back. Ata Mukwena, he's my technician focusing only on COVID work, with generating some very good results in that space. Next. So this is what he is doing, so to just keep pressing, going forward. I'm just trying to explain the steps he is using in doing this work. But the people who led us to believe that we have something were our colleagues in the US. Next. And this is the assay that we're using and comparing with antibodies that we know the Delta variant and all those things. That's there. So I'm not going to share with you the results, so to say, but we're on the right track in terms of this. Our intention, because COVID is a very interesting condition, is that we have people who get infected, walk around, don't know that they have the disease. You have people who get a mild disease. They need to stay away 7 days, 10 days, and recover. They might use concoctions or other things in that period. Then you got those who get sick, who need to be hospitalized. The fourth group that knew is the long COVID, the sequelae. So we still need to do, even someone says the COVID is gone, but the long COVID is still with us. People need to go. So as we do this work on COVID, we are looking at that, finding solutions for those people. I did not need to present hard scientific data, but I wanted to tell the story that part of our journey has been enabled by collaborating across and this. And the last work I just want to share briefly is the fact that since the Madagascar saga, WHO had to step in, bring us scientists and experts to advise countries about how to do high quality science around traditional medicine, because if it's bad science, it continues to denigrate traditional medicine. And so if we do high quality science, so we developed clinical protocols, we developed what to do preclinically and to do this. So we now that the doors opened, we started to visit countries. I think about 12 that it was tiring. But it was worthwhile because we started to begin to see where does expertise lie, where there's the best infrastructure, including manufacturing, because we have to produce this, not just for your country, but you have to produce for the continent, because we have a free trade zone now. So we needed to make sure that we have facilities that can manufacture good manufacturing practices, export these across, involve the regulatory authorities in the speed of approving these studies and registering these products such that they can be made available. And so that's the part of work. In that process, we creating bridges. So we see one of the things with COVID was that Africa had low COVID numbers. So if you want to do a clinical trial to recruit 3,000, you won't make it. But we can now do multi-center, multi-country studies to do this. And we had teams in Uganda, elsewhere, of doctors who were committed to doing this, and which was good. Very good for me, because one of my problems in South Africa, when we want to do clinical trials, I've been battling to find African doctors to lead a clinical trial on African traditional medicine. Funny enough, I've always been finding white doctors. I don't know why. And we bring them to work with healers. We bring them to work with traditional healers. We don't put healers aside. They are always integrated in the projects that we do. This is their herb, it's not mine. And they have to be part of it. We publish with them. They will patent for them. It's their IP, it's their knowledge. We're not crooks. We don't steal. I must say that. And so one thing I like about this, God just blessed us with partners who respect indigenous knowledge, not wolves. Not that we chose them, but we just found each other. And so we don't have battles about this. And it's been good for our students. As told you of Nonhlanhla, I went to train. It's been good for our students for capacity building and the sharing. And one of the things we're doing now is to bring these technologies here such that the work can be done here. We don't need to send extracts elsewhere. And some people thinking, hey, they will steal. That's not. But one thing is that even though we found problems within our borders, like I'm saying, our regulators and other things, we are finding that we can do this work in other countries. So if South Africa gives us a problem, we'll do a clinical trial in Nigeria. We'll do it in Uganda. We'll do it elsewhere, because Africa is one. We can do this. And the thing is that the results, when you partner like this, come faster. And lastly, is that the benefit is for everyone. And by that, thank you. Next.
Thank you, Prof. Thank you, Prof. Gqaleni. I did say that you'd be inspired by him, you'd be charged, you would find yourself challenged to change the way you think about doing research as an African in Africa. In my language, if you are truly excellent in whatever you do, there's a way of calling a person. So, Njebengu professor, I would say Professor Pro. If I teach, I would teach at Professor Pro. And quite a gift to our university and to our nation. As personally challenged me as a social scientist, but how do I change the way I do research so that I live out? I talk a lot about decolonization, but how does it start emerging? How do I make sure that it starts emerging in the way I do research, the research questions that I ask? So I'm going to open the floor now so that we take questions from Prof. Gqaleni. I think we went a little bit over time, Prof, but I thought I'm not going to stop you. I could listen to you forever, and I'm sure everyone else could. There's something I was saying to a friend of mine the other day, that, you know, this obsession with sticking to time during important gatherings is so un-African. Tina is our pela. It will finish when it's done. When we have said everything we need to say, it doesn't, you know, so I'm going to adopt a more African way of doing, of sharing. So I allowed the Prof to go over time, but I think we can give, it's 12:03. Could we give 15 minutes for questions? 50, there's too much. Okay, so we have 10. We'll give 10. Mrs. Zondo is not, the Zondo Commission is not on the same page as me. Okay, so I've seen hands. I think, Prof, you were the first. So I'll take Prof and the second person right behind, so that's the second person. Then we have someone. Okay, thank you.
Thank you so much, Prof. And I think I'm glad that it's a social scientist that's also asking the question first. One of the reasons why I have been very reluctant many years ago, as far as our engagement with the West or the global North, is this issue is the issue of unequal engagement. And you find this, and I really applaud what you're doing, and I'm happy that we've come this far, because we cannot Africanize, we cannot decolonize if we don't bring our ways, our ontology and epistemology to the fore, okay? But the challenge I'm finding here, and I didn't want to speak, but I say if I don't speak, I'll be grumbling if I get out of here. It's the fact that we have to now allow the West to legitimize what we do. And that is problematic for me in our engagement. Because I heard you clearly saying a WHO had to send scientists to guide us so that we don't practice bad science. And that's for me, we cannot continue to stoop low, we cannot continue to bow. What we do have scientists here. You are one of them. What makes it difficult for you and your colleagues to show the West how it's done? Why can't WHO listen to us totally? Must they send their experts here before the science that we produce will be legitimized? I think I have a problem with that. And until we are able to negotiate as equal partners. I'm telling you this because personally, I'm not going to engage with any institution. I don't care who they are in the West. I'm not prepared to collaborate with them unless we agree we are equal partners. I mean, from conceptualization to dissemination. If it's not on equal basis, then it's not going to work for me. For us to actually promote indigenous knowledge, which is what you are doing, and I'm so grateful to for the work that you do. We must get to the point where we cannot allow them to, we cannot authenticize what we do from them. They are not, who said that they are the one who practice right science and then what we do is bad science? Does it mean that our ancestors, our forefathers practiced bad science? How is it that they were able to cure and they continue to cure people? So I think for me, until we are able to answer that, I still have a problem. I have a problem with that kind of engagement. Thank you. I'm sorry for taking a lot of the time.
Yeah, I think you're as angry as I am on this subject. So, but let me just assure you, maybe I should have qualified WHO Afro. This was being led from Brazzaville using us African scientists from Africa. So it was our peer review mechanism to ensure, and we did site visits. It was us African scientists, even coming to South Africa, because this is our IP, it's our intellectual property. We need to safeguard it, but at the same time, we need not want our public to be shortchanged, and that's what we do. So please be assured. Thank you.
So much. So I'm going to take only the two other hands that went up because otherwise we'll run out of time. So there was someone, yes.
Thank you very much, Chair. My name is Kukuma Zibugo from African Languages. Thank you very much, Prof, for the wonderful presentation. We have been patiently waiting for the end product of your research. The question that I would like to ask, Prof, is at the end of the entire research, how are you going to name the end products? Are you going to use the fancy scientific terms or indigenous names? Thank you.
Thank you. I should say a question to Professor Pro. You know, I always intrigued by the name for professor in isiXhosa, Unjingalwazi, which means someone who knows everything. Yeah. So I'm asking it in that context. My name is Laverne Samuels. I'm the Director of International Education and Partnerships at Durban University of Technology. But my background is also in the health sciences. So this is an area that I'm also very passionate about. And we've been driving decolonization, social justice, epistemic justice, which I think this is very much about. But with all these opportunities to indigenize knowledge, but more importantly, take indigenous knowledge to the global stage, to what extent is it being explored? For example, you've had this project, but I think there's also that this is just the tip of the iceberg in terms of what could potentially be taken to the global stage. I'd like you to comment on what it is that universities, government, institutions can do to take this project to scale and not to have it as a nice to have in some corner of the university, but to help it define our academic project. Thank you.
Thank you. Yeah, difficult questions. Sisi, igama leyeza sizolithola kubaba uNgabinde. Because lelakhe, it's just now in the course of doing research, we've coined it Product Ngabinde to still retain the sense of ownership and honor to him, the old man. And so that's what we will do. I hope you're happy for that. Thank you. So what he calls it will be, but we can brand it, it's not a problem. In terms of marketing principles, it's not a problem. Thank you for your question. Look, one university that has impressed me, the Simon Fraser University in Canada, in terms of its approach to community engagement, to public engagement. It's a very engaged university. We linked with them with their indigenous communities. And so we have projects that we're doing with them, with the indigenous people of Canada. We took our traditional healers to that university. We had rituals to welcome us. That was amazing. We went to their villages, to where they work. We met with their traditional healers. We had an exchange. They came here with their chief here to Durban. The same healers welcomed them here. We have had discourses. We've written now, we're competing for one of the, in Canada's terms, it's probably one of their top tier grants. It's a 7-year grants. They call it the Frontiers. And it's about indigenous knowledge. And you will be amazed. We have been left probably with about 20 that were shortlisted to go for the final round. Whittling. But what helped us to be shortlisted, I think, because the reality of climate change and the whittling of indigenous knowledge became palpable in Canada, and such that they do realize that. We're hopeful that we'll get it. And I think some of the work we're going to be doing through that grant is going to be really groundbreaking. But it's also bringing these voices of indigenous people, our indigenous healers of KwaZulu-Natal and Vancouver and Canada working together in this thing. It's a process because skeptics are there, and so we have to always step up in terms of generating evidence to say we're not talking mambo jumbo. The things we're doing are serious to be noticed. It's also about these universities we're part of. It's a battle to get a module, our curriculum on traditional medicine. We have a module generated by us through WHO Afro for health science students. But our curriculum is so cramped. We started in 2000 to train our medical students on traditional medicine. Quickly, our colleagues changed the curriculum from 6 years problem-based to 5-year program. What went out the window first was traditional medicine. Cut out, you see. And these things, to negotiate that back into the curriculum now, they went back to 6 years, but it's no longer traditional medicine that finds its way back. Nursing is the same, the curriculum is full. It's difficult to find. So we have to kind of now navigate our way through these spaces. When we take pharmacy students, they come to my laboratory. The shock is, they come on their 4th year. The first shock is, I never knew there's a top-class lab for traditional medicine in this university. In my 4th year, as I'm leaving, we give them a lecture, and they're like, I've never heard all of what you're talking about, if you understand. I tell them, the Greeks told you you are 3 in 1, mind, body, and soul. Our own epistemology says we are 7 in 1. And I tell them what components of their existential reality. They're like, I never had this. And my 4th year, I'm leaving the university. So we have this dilemma that we're trying to battle. So we've got to negotiate to Africanize our curriculum in Africa. Strange. You know, our regulatory, these councils, you wonder, where do they come from Mars that they have to tell a medical student training in South Africa today, they cannot learn African epistemologies, African ways of knowing, understanding health and disease. Strange. Thank you.
Thank you, thank you. I think that's a perfect note to end on. And I think the charge really is on us as the academic community. We have to find ways to legitimize our African ways of being and doing. Prof. Vivian was talking about, needing to be legitimized outside, beyond our borders. We are the problem. We illegitimize our ways of being and doing. The struggles that you've had, you wouldn't have had them. So I think that's really the charge about what are we going to do in our individual spaces as African academics to change, because how we think about knowledge needs to change. Thank you so much.